Hello everyone. Thank you for joining us today in the 18th lecture of the Biology webinar series. I am Shinjini and I will be your moderator for today's session by Dr. Sudeshna Fisch. As many of you know, we've been hosting these lectures, fortnightly lectures, over the past months to keep us stimulated in these dire times, with Saturday lectures on topics related to other disciplines and Sunday lectures on different topics of life sciences. Before we begin, I would like to remind you that you can post your questions in the YouTube chat box and we shall moderate them at the end of the lecture, and a feedback link will be provided in the chat box near the end of the session and it will not be sent by email. I also request you to please be a little patient regarding the issuance of the certificates and you shall receive them by the end of the week. So without further delay, our mentor and convener of the series, Professor Reena Ray Banerjee of the Department of Zoology, University of Calcutta, is unable to join us today, but she was looking forward to interacting with our speaker and she sends her best wishes for today. So I would like to take this opportunity to welcome our speaker for today, Dr. Sudeshna Fisch. A very warm welcome to you, ma'am. Ma'am
Thank
was
you.
unable to join, but she
That's
sends
okay.
her wishes for you. So Dr. Sudeshna Fisch is Associate Director of a busy and prolific preclinical core, the Cardiovascular Physiology in the Department of Medicine of the Brigham and Women's Hospital, Harvard Medical School. Dr. Fisch's research focuses on preclinical therapeutic studies using mouse, rat, and zebrafish models, surgical and non-surgical techniques, including advanced cardiac imaging and data analytics. Her prior work includes intellectual property, research commercialization and grant funding strategies, with stints at the Broad Institute of Chemical Biology, Nanobiosim in Nanotechnology, Harvard's Office of Technology Development, Partners Office for Research Ventures and Licensing, and Fisch and Richardson LLP. She was born in Calcutta, India, earning her BS and MS degrees in zoology and genetics from the University of Calcutta, where she received National Merit Scholarship awards. Dr. Fisch received her PhD in biological sciences on a full scholarship from Bowling Green State University, Ohio, before moving to Harvard Medical School for postdoctoral work. Dr. Fisch's publication list can be found in the Harvard Catalyst profile page, and she was recently selected as a brand ambassador for Harvard Catalyst's clinical and translational training and educational programs. She also serves as guest editor and reviewer for scientific journals and is always seeking new collaborations and learning opportunities. So with that, I would like to invite ma'am to please, you can start.
Yeah, sure. Thank you, Shinjini. And thank you, everybody. And thank you, Dr. Banerjee. That's Enadi for me, because we go back a long way to Gokhale Memorial Girls School. And thanks for the invitation. And I'm really delighted to give this talk. And I'd like to acknowledge that some of the work that are on these slides are from my, more basic aspect of the translational work that I do, but I will touch upon many of the important thinking around translational science that is currently ongoing both in my research laboratory, which happens to be a research core and serves a very important tool for the research community here in the US and also globally. We have collaborators across, but also drill down, actually talk about how you get to that translation, how you do clinically translate some of the very basic bench side, sort of research that we do as PhD scientists, because here in the US, we work with both PhD and MDs, and in some cases, JDs, the lawyers who write the patents, and we all come together and basically figure out where the most translatable value is. With that, I'll just get started on my talk. I'm going to need just a second to go full screen, and for those hold on just a second. I just want to make sure that my, it doesn't matter. So I will be on screen, but I wanted to hide my camera. Anyway, I'm starting now. All right, so the title of my talk, as you can tell, is I'm going to mostly talk about the preclinical modeling. And it's a challenge sometimes to come up with really good preclinical models that mimic human disease. So, I'd like to start by putting in a plug for the core I run. The director is Dr. Liao. She's actually at Stanford. And, I happen to be at Harvard, so it's a cross-continental collaboration model. But we have a ton of different options within our core. We have like the imaging tool, which you see on the left, where we can basically non-invasively study disease and study disease progression. And we can come up with, plans for how we're going to treat the multiple species or models using surgery, using injection, using ex vivo, using cell culture, and even be able to do blood pressure. So, this is the sort of typical research core model that any big institution in the US has to kind of come up with the tools and strategies to help the research community perform some of the academic research that then goes on into the industry partnership to be translated into products that are used in therapy. So as you can see here, there are several ways you can approach a disease. The disorder could be any type. And the left panel, you see, like I have liver disorder. Let me see if I can do the laser pointer. So I have a liver disorder, heart disorder, kidney disorder, you name your favorite disorder. And then you can actually use these various approaches from human genetics to proteomic, metabolic, radiomics or imaging, or chemical genomics, meaning using small molecules, and a combination of genomics, your genomics information from the human population to study your favorite disorder. You can do it from soup to nuts, from preclinical modeling all the way to translation to the human trials. And so there are several advances that have led to this point. So one of the ways I like to think about this paradigm is, we first scientists have to identify a disorder, your favorite disorder, and maybe come up with try to think about is there a genetic basis for that? And where is the need? And I think with those two things, you have to poise yourself to understand that to translate human genetics, you need to go through some basic steps where you establish that they're safe and efficacious before you introduce to human body. So you also, in order to get to that point, you also need to develop cell and biochemical assays, which are in vitro assays, not necessarily preclinical, but in vitro assays. You can to test your model, to test your assumptions. You need to go through that. These are necessary steps for you to translate a model. Finally, of course, the goal being discover therapeutics, discover pathways, and then ultimately to make very informed clinical endpoint decisions and surrogates for further in vivo testing, perhaps through clinical trials. So for today's talk's purposes, I'd like to focus mainly on the transcriptional and the post-transcriptional mechanisms, how these can be used as potential targets for drug discovery in cardiovascular disorders. So like I said, one of the approaches could be genetics, chemical genomics, but the goal would be to find maybe novel therapeutics and biomarkers. In the context of cardiovascular diseases, I basically just like to silo them into some of the models that I am most involved in right now. On the left, you have the heart failure model, and I don't know to what extent the audience is a heart focused, but essentially heart failure can be for many reasons. The underlying etiology of heart failure is dynamic, it's heterogeneous. You could have a heart failure form where you have a heart can't fill. It cannot fill blood enough, so that because it has very thick chambers that have been caused by fibrosis or some other reason, but the function is perturbed. And then the other model you can have, the heart can't pump, meaning the heart can fill, but because it's so sort of the stretched and thin, the chambers, it can't really contract. So these different conditions that cause the heart to behave different ways lead to the heterogeneity of heart failure. So you can have what is known as the HFpEF, which is becoming increasingly commonly seen in the patient population, and particularly in some cases in menopausal women. And then you have the HFrEF. So if HFpEF is not treated, over time, it may develop into reduced ejection fraction, or HFrEF. You have the coronary artery disease, which is the number one, single disease risk factor for, any of the heart attack or embolism that we see in the American population. It can be caused by lesions or plaques that eventually can rupture and cause aneurysm. So coronary artery disease is a serious complication that can also lead to heart failure. Then you have arrhythmia, which is, basically it's a heart block, or, it can be noticed in an abnormal ECG pattern. And, in a normal heart, you have certain different conduction rhythm, and that gets aberrant. And there could be many causes of that. So it's important to understand that there are different silos within cardiovascular disorder. And then we have diabetes, diabetes is significantly now, one of the major causes of cardiovascular disease condition. And so one of the causes of that is dyslipidemia, like an aberrant lipid lipogenesis or lipogenolysis. Obesity is a major risk factor, and metabolic syndrome. And ultimately, if you leave this untreated, you can cause all of these other issues. So that sort of sets the stage for my next few slides. So cardiovascular disease, we know the incidence rate is it's forever rising. Now with COVID, the complications of cardiovascular disease is even more dire, because ultimately the ACE inhibitors are ubiquitously expressed in the heart and the lungs. So ultimately, vascular diseases will affect a large number of other diseases also. So it's important to think about cardiac diseases and also a vascular disease. At a very basic level, to understand that heart can become misshapen, like, normal physiology, a heart looks a certain way, but in disease state, the heart can also look a different way. Now, there can be two forms of heart diseases. One is the concentric form, which is where the heart wall, especially the left ventricular wall, gets very, very thick. And that's when the heart is compensating for the load that's on it. And that's called concentric form, a form of hypertrophy. And then you have on the right, what you see is the heart wall, the ventricular wall is thinned out. In the middle, if you have a normal heart, you see on the side, you see a thinned wall of the LV. What happens in eccentric form is the myocytes, which are the primary functional cell type in the heart, they start to apoptose. And so this apoptotic phase, it ultimately leads to necrosis. But either way, the concentric or the eccentric are the two forms of hypertrophy that one needs to understand at a basic fundamental level to understand cardiovascular disease therapy. Of course, we all know the statins are the super drugs, they're the major line of drugs in the market, but mechanistic understanding and the mechanism of action and all the implications of what happens when you use statin is still poorly understood. My goal in this is to essentially talk about transcription and a little bit of post-transcriptional mechanism. And I don't think I need to go over these basic premises that most of us learn in our MSCs and BSCs, is that how transcription occurs. But the important thing is you go from DNA to pre-mRNA to RNA, and the RNA, the mRNA, the messenger RNA, is actually a very critical player in a lot of the dynamics that go on inside a cell, right? So, and the messenger RNA is what codes a protein, and ultimately we have protein, right? That also functions in the cells. But you can have mRNAs that are fully functioning. You can have now, we know that there are microRNAs that regulate both the functions of proteins and mRNAs. So there's all this heterogeneity and dynamic going on inside the cell. So this this kind of dogma that we held, that DNA to RNA to protein, and being the protein being the main functional factor, it's no longer true. And so, transcription remains as a key point in most of disease research that I do still. The way to understand transcription is that, you can have either a repressor or an activator. A repressor, which is a molecule, which is an mRNA that turns a gene off and then turns sort of a chromatin keeps a chromatin into a condensed condition. And then you can have an activator, which turns it on. And then there are various mediators that do this repressor and activator action. And it's extremely important to realize that you have a complicated play of transcription factors, RNA polymerase, and not to forget the actual methylation and the deacetylation and acetylation and all the other marks that are on the chromatin that have to play a combined role with the RNA, with the protein, and the epigenetic regulators to turn a gene on or off. And this turning the gene on or off is critical in understanding how a disease progresses. Although a gene may be disease may be multifactorial, multiple genes are involved, it's still important to realize that there are repressors and activators of certain genes that are still in play in most of the major diseases. One of the groups of genes that I've been always interested in is Krupel. The Krupels are, these are DNA binding transcription factors. They have many different things. There are right now about 18 of them. They regulate a whole broad swath of cellular work. They regulate cell growth, differentiation, function. There are several isoforms, KLF1, 2, 4, and 5. And the difference lies mainly in their fact that if you look at them, they all have the DNA binding domain, these zinc finger binding domains. But what they have, you see there have some of them, for example, KLF2, which is something that I will talk about today, has both a transactivation domain and a transrepression domain. And the transactivation and transrepression domain together, that means that this particular molecule is able to both bind the side of a chromatin and then activate a gene that's downstream. And in a certain context, it may be able to repress a gene and turn on, repress a gene and not use its transactivator function, but you can act as a dual player. So that makes it a very interesting molecule to study. So you can see the differences in some of these molecules that I've shown here are in their ability to activate or repress. This is sort of showing you a sort of like a domain structure, like a structural information on what the difference between each is. And I just wanted to focus on KLF2 and KLF15 today. And what you see here is that the KLF15 structure is a little bit different from the KLF2 structure. So you have the transactivation domain and the transrepression domain next to each other, juxtaposed in KLF2 molecule. And in the KLF15, what you see is that the smaller transactivation domain, a bigger repression domain. And then you have this DNA binding domain right here. That is constant among all of this. So that means these transcription factors bind DNA and then do their function, okay? That is a necessary function for them to bind in order for them to perform their own function. So the thing about Krupels is that it's expressed, it's an interesting set of players for me because it's expressed in a whole variety of cells, of cell types. It's expressed in the heart, the vessel, as you can see, it's also a major player in metabolism. So it's expressed in adipocytes, which are the lipid cells. It's expressed in macrophages, immune cells, stem cells. And it's got a new and upcoming role in cancer biology and aging. So it's ubiquitous. These are important groups of transcription factors. So far, there have been SNPs, which are the variants that are identified in human genome that can either be very rare or can be very common. And it turns out that the rare SNPs are sometimes often more associated with the disease state. And coronary artery disease, which is something that I mentioned before, notice that KLF15, which is one of the zinc finger domain containing Kruppel-like factors I showed earlier, a SNP was identified that is suggested as the risk allele for coronary artery disease. And there were studies, clinical studies, showing that about 1,100 families or with early onset CAD and 1,300 individuals with or without CAD from a cath lab. I mean, so there's direct association of this SNP with an actual need for doing catheterization later, right? So it's an important point to note in its translation potential. If you look at the tissue expression of, say, Kela-15 mRNA, that's another important thing. If you're doing a preclinical model, maybe early on you can do some assessment to look at a particular gene, if that's what you're looking at, to look at, say, where is it expressed in a broad swath of tissue. You can take mouse heart, you can take rat heart. For the most part, they will show you, the expression level that you need to see. But essentially what you see here in that Kela-15, what I noticed early was that it's expressed in liver and kidney. And actually, this is all work published in PNAS. This is work done by me, so it's all good. But you can see that the level of expression in the heart is pretty strong. So is it expressed in the liver and kidney, but it's not expressed everywhere. So that's actually good because we need to come up with a molecule or a target. That's important that it's not, if you were to target a particular protein with your therapy, and if it's everywhere, you're going to hit it everywhere in all your cells. But sometimes it's important to know if it's expressed in certain tissues and not others. That's actually an important point, that it might help you to target certain diseases and not others. So that's a good point for you to remember. Looking at the cellular expression of Kela-15, you see that it's exposed in all these different cells, right? This is using LAG-Z staining, which is very common to do, but you basically need to show that not just at the tissue level, but you're also expressed at the cellular level. So it's expressed in adipose cells, skeletal muscle, heart. You can see how clearly it's in the myocytes and around the lining of the blood vessels. So it's all important, good sign that Kela-15 is a good transcription factor for us to go for to see whether or not it affects disease. So as I said, Kela-15 in the heart and the vessel, and also in Kela-15 liver metabolism. So I'll talk a little bit about these two. Expression in the heart. So we notice that postnatally, like after a mouse is born, and in this case on the left is the mouse and the rat, what I notice is that Kela-15 expression at the RNA level, and by the way, for those who don't look at these routinely, these are just northern blots. They're looking at levels of expression of these RNA using a very standard technique that we don't really use that much anymore. We mostly use qPCR, but at the time that I did this, it was important that we showed that these were indeed expressed. So Kela-15, as you notice, post 3 days after birth is really not there. And by 15 days, it's sort of coming up. And then by 56 days, it's really strongly expressed in a postnatal mouse heart, right? But and actually, along with that, you also see a possible target for Kela-15, which is now not a possible target. It is a target for Kela-15. A downstream target is also expressed. So the correlation of the expression of GLUT4, which is a glucose transporter protein, is downstream of Kela-15. And that's an important element that you need to look at when you're looking for expression level changes. Now, if a particular RNA or protein is expressed in a certain phase of the life, for example, in this case, we see that it's induced postnatally, it might be important to think what other genes are also expressed in the heart at the same time. So when we look at that, what we see is that ANF, BNP, which are commonly used as surrogate endpoints for many of the cardiovascular therapeutics, and these are these proteins that are secreted, and they are BNP is very specific for left ventricle, and ANF is for the atrial segment. These proteins, they are hallmark features of cardiovascular disease models. So we noticed that in the rat heart, also ANF and BNP were upregulated. So you can see the opposing relationship between ANF, BNP going up at 3 days and KLF15 and GLUT4 down. And by about, postnatally, about adulthood, KLF15 goes up and ANF, BNP are down. So that's important to note, because this is how you think about your study, like what goes up, when, and then what goes down, when. Postnatally, meaning after birth, you see something. Okay, what does it really mean? So we can study that further in the disease context by inducing, remember what I described as eccentric and concentric, you can induce a form of hypertrophy in rats or mice by doing a surgical procedure known as banding. That's very typical, and I'll talk about it in my next slide. What you notice here, and this is an important slide, I want to spend a few minutes on it, because this sort of sets up the stage, how you want to do a study. What is induced postnatally, you see that following banding, sham means no surgery, there was some, like some opening of the chest, but closing the chest. And in banding, following banding, KLF15 and GLUT4 both go down. Whereas following sham, the levels are pretty standard. These were done in adult mouse, so KLF15 levels were high, and then following banding, they go down. 4, 5, 6, and 7 are just unique technical replicates. And that's important to do for any study you do. You want to have enough technical replicates. In the rat study, you see that following doing the banding, the ASC, the KLF15 goes down, and ANF, BNP, as you expect in disease, they go up. Now that is very important. So KLF15 is reduced in hypertrophy, ANF, BNP, as expected, go up. That's an important observation. So we started thinking about, okay, well, what does it really mean clinically? You look at the aortic stenosis model of CABG patients from the Framingham Heart Study, and we see that KLF15 expression is very low in the aortic stenosis patients. And the KLF15 mouse heart is dilated. So the CABG patients, that you can see dilated heart, and we see that in mouse. And then also the staining in the human tissue correlates with what we saw in the mice. And this is sort of the normal human normal human physiology, where you expect KLF15 to coincide in the cardiomyocytes. So looking at this, we find that there's a correlation between the disease heart in mice and man. And that's important, because you want to have at least a couple of different models before you go to the human stage and say, hey, my gene is really important because I know that this is implicated in hypertrophy. So we generated a KLF15 knockout mouse. One of the ways you can establish that the gene is necessary and sufficient is that you can knock it out, right? And I don't want to, and this is all published data, so you can look it up on the technique of how we knocked it out, but it was a PGK neomycin cassette put in. It's a systemic knockout. So once you knock out the gene, then you ask the question, well, what would happen if we take these mice and we subject them to different kinds of stimulus? Like, how about if we do pressure overload, meaning we put a lot of pressure on the heart, such as by banding, which is something that I talked about before. Or you can add different agonists like phenylephrine and things that induce hypertrophy in the heart and things that induce hypertension in the heart. This is a very basic way one can ask a question about an importance of a gene. And then, we ask these questions in the context of, well, pathologically, what can you do? Like pressure overload, PE stimulation, Ang2 stimulation, endothelin-1 stimulation. Okay, you can do those both at the cellular level. And then you can also do it by infusion, if you have that approach. And you then look at, well, what is the hypertrophic gene program look like under these conditions? We expect that ANF and BNP, as I discussed, to go up, and SERCA, which is a calcium handling protein, generally tends to go down. Also, another very important thing to remember, and at least for the cardiovascular people here, is that there's a switch of the beta to alpha isoform that is very typical of postnatal birth, and the same thing happens in heart failure. So these are sort of critical paradigms that's existing in the hypertrophic gene program, and plug that in to your KLF15 or your favorite gene model and say, hey, what do we see? In normal pathologic hypertrophy, we see the cardiomyocytes, which are the singular most important cell type functionally for the heart. Cell size increases, protein synthesis increases, and you see big sarcomeric changes. These are the expectations, right, of hypertrophy, if your model is correct. So we did the pressure overload. We noticed that when we did a severe binding, which is so essentially this is the aorta and this is transaortic constriction, and this is where you tie the band for aortic constriction, ascending aortic constriction. This creates a tremendous amount of pressure on the heart. When we did that as a very high gauge, we noticed that we had a ton of death, 100% mortality. So we reduce the amount of our overload, and you can do that. That's similar to adjusting dose of your drug, right? You adjust the dose of your pressure. And you know to create a mild phenotype, and you see that the majority of them survive. And this is a typical Kaplan-Meier survival curve, where you see that the days after the AAC, which is ascending aortic constriction, the KLF15 knockout mice, they die, but they kind of stabilize. So there's some survival. So then we started looking at all the things that I talked about: gene expression, cardiac function, mechanism of function. And what you see here is that KLF15 is indeed reduced with cardiac hypertrophy, as we saw in the AAC model, in the mouse that I just showed you. That this is actually. I apologize because I think this is sort of a repeat of a previous slide. But essentially, what you see is KLF15 is reduced to hypertrophy, which is something we noticed. And the question we ask next is KLF15 affect hypertrophy? So when you knock it out, that's the question you're asking. You know, how does it affect? So if all of these negatively regulate KLF15, and do they turn on the hypertrophic gene program? And then how you affect this, right? Okay, so what we noticed is that in the knockout model, we noticed that when you knock out KLF15, you have all these overexpression of ANP, BNP. But when you overexpress KLF15, you notice that you can reduce ANF, BNP. And these are reported gene constructs where we had basically looked at the upstream, the promoter region of ANF and BNP, thinking about mechanism that does KLF15 actually directly affect ANF, BNP. Is that a fact? Is that one of the mechanisms that's happening here? Why KLF15 is a major player in this form of heart failure? Is that what we notice is that when you knock down KLF15, actually when you overexpress KLF15, you see in absence of PE, then you have a very low expression, you know, of ANF. But then when you overexpress KLF15, you see that you're able to inhibit the ANF promoter. So it's if you compare EV with EV, K15 with K15, I apologize because it's been a while since I presented this slide, but essentially what you're looking for is KLF15 is able to inhibit the ANF expression and BNP expression. When you overexpress KLF15, you are able to do that. And we show that the leucine incorporation is an indicator for protein synthesis, and then fold change in the geometric mean of the sizes of the cells. And what you see is that in presence of phenylephrine, which is a neurohormonal agonist that causes hypertrophy, KLF15 is able to reverse the hypertrophic phenotype. So you have smaller myocytes here, and you have large myocytes here. So this is in presence of PE, and this is when you overexpress KLF15. Right? So all of this indicates that KLF15 inhibits myocyte, meaning cellular hypertrophy. And you see that also under the scope that KLF15 null mice themselves are actually, these are the null mice that are bigger than the normal KLF mice, suggesting that there's also an effect on the size and the hypertrophy of the myocytes when you knock out KLF15. So you look at both overexpression of KLF15 and you look at the KLF null mice. The most important thing that I noticed was that the KLF15 knockout in presence of banding, they showed a functional difference. And actually, that's very important to know. Remember what I talked about, the eccentric form of hypertrophy, is that an eccentric form of hypertrophy, what you expect is a thinning of the wall. So that this line between the green and so this is a picture of an echocardiogram. For those who are familiar with it, this is an old machine, but now we have like a state-of-the-art 3100. The pictures are much better. But essentially, what you look at this, the wall is much more thinned out in the KLA15 null mice, and suggesting that they have an eccentric form of hypertrophy when you don't have KLA15 systemically, right? And when you have a heart rate to body weight increase and a LV dimensional increase, those are the structural landmarks that tell you that, yes, you have a functional problem. And most clinicians, if there's any in the thing, would know that that is the one that directly relates to a functional difference in the heart, which is known as ejection fraction. So this is a typical clinical view of a heart. If you look at a cross-section of the same hearts that I showed you in an earlier picture, you see this is dilated in the KLA15 null mice, and fractional shortening, which is just another functional parameter, you see severely affected in the KLA15 null mice, meaning functionally these mice are really poorly behaved. And that can explain why we had so much death when we put more pressure on them. So when you put stress on an already stressed heart, that's what you create. You cause so much stress that the mice cannot survive. We also noticed that there was enhanced fibrosis in the heart tissue, and that's a critical hallmark of disease state. So that's important to note that KLA15 also affects fibrosis, not just hypertrophy, but also fibrosis. Mechanistically, there's so much we can talk about. By looking at this picture, you can understand how busy it is. But if I focus just on this pathway, the phenylephrine Ang2 pathway, you see how there's a downstream cascade that all leads up to this key transcriptional players that can sit in some kind of a gene promoter upstream in concert with HDACs or HATs, which are the epigenetic regulators, and they regulate hypertrophy. There's physiological hypertrophy and there's pathological hypertrophy. So if you look at this picture, we want to see where KLA15 rests. And our goal was to establish what other players are playing, are connected with KLA15 to be able to do what it does. So it turns out KLA15 negatively regulates this picture, because MEF2, NFAT, GATA4, these are all pro-hypertrophic players. And KLA15 negatively regulates this to affect the outcome in pathologic hypertrophy. So when you knock KLA15 down, these players are somehow involved in developing the hypertrophy and leads the mice to die, to heart failure. So we looked at the expression. Well, so to test the hypothesis, you always look for, either do Western or qPCR and to figure out, if your hypothesis is true. What we noticed is that none of the expression of the players that we thought were involved were really changed. There was some indication at some point that there was a MEF2 and an NFAT connection, but and that became a very interesting story in itself later on. But in the context of what we were looking at here in the KLA15 knockouts, we didn't see any big change in expression. So then the question we had to ask, well, if the expression didn't change, what else? How else could they have affected ANF-BNP? It turns out MEF2 and GATA also sit on the promoter of ANF-BNP. And what we did a reporter construct assay. What we noticed is that in presence of MEF2 and KLA15, this MEF2-induced expression of ANP was reduced, meaning there's a synergism between, antagonistic synergism between MEF2A and KLA15. And the same we noted for GATA4, meaning in MEF2A does what GATA4 does to BNP-NPs, turn it up. And when in presence of KLA15, when you add KLA15 in, it brings it down. So that's important to kind of establish how this is going on. The NFAT story is kind of similar, but it's a little bit more complicated, but there is some flair from some role of NFAT-C involvement also, as you can tell here. And that's a calcium story, and that's a very interesting story in itself. I can discuss another time. These are, so basically all we are establishing here is KLF15 inhibits GATA4, MEF2, and FAT-mediated transcriptional activity. How does it do that? We know that it has zinc finger binding proteins, a zinc finger binding domain. So essentially what we did was known as EMSA. It's a electromobility shift assay. This is very old school assay that we did at that time, where we had created we had like overexpression of KLF15 or like add GFP, and then we basically have a small segment of a promoter that we bind these particular molecules on the left to, and we notice a shift in the binding, and the shift in the binding suggests that something is binding to that promoter region. And what we noticed that there was a shift in binding using MEF2 that was mediated particularly in the condition of AAC in the KLF15 null mice, meaning KLF15 null mice that underwent banding showed an increase of the MEF2 binding at the ANF site. Now that's very important, because that also suggests to us that the MEF2 binding is enhanced in absence of KLF15, especially in stress. Same story for GATA4. Again, we're establishing context and mechanism. So, summary, KLF15 expressed in monocytes, overexpression inhibits cellular hypertrophy. In cardiofibroblasts, we showed that it inhibits cellular proliferation, pro-fibrotic gene expression such as CTGF. The KLF15 null mice, which is very important, that exhibit the eccentric form of cardiac hypertrophy, and it inhibits several key hypertrophic transcription regulators. Now, I'm going to switch gears a little bit, and for the time, I wanted to talk about KLF2, so I'd like to go to KLF2 directly, but I'd just like to give you an overarching view of where the KLFs are right now in the big scheme of things. I mean, the field has expanded rapidly since my last, foray in it. And Dr. Jain, who's my postdoc advisor, he's a National Academy Science member. This is a slide that I borrowed from him because I wanted to show you that from the time that I was involved in this field, the Kruppel-like factor 15 has now grown and encompassed a whole array of disease spectrum. And essentially, it's what's now held, known very clearly is that it's particularly important in differentiation activation. They're master regulators of the endothelium, essential regulators of innate adaptive immunity, and there's SNPs identified, a lot of them now in anemia, cancer, pulmonary hypertension, and atherosclerosis. And so it's important, and actually this is also published in ATVB, so one can look this up. But it's important to recognize that what we thought originally for a KLF was, we're going to look for the role of KLFs and, the heart and see where it goes. Now the field has mushroomed a lot. I don't want to dwell too much on the KLF15 and the vascular smooth muscle cells, but it suffices to know that when you have a clogged artery, I mean, that's coronary artery disease for you, right? So if you can find a player that can mitigate the clogging of the artery or somehow prevent the proliferation that leads to the migration of the smooth muscle cells and prevent plaque rupture, that should be an important pathway for one to look at. So now, again, just like the cardiomyocytes that I just talked about, in smooth muscle cells, you can also have physiologic and pathologic hypertrophy, and each is accompanied by different states, and that lead to either physiologic growth, where the cells are differentiated, meaning they do not proliferate, they're quiescent, and they contract, as opposed to pathologic occlusive disease, where the cells proliferate, they migrate, and they extend the ECM, which is the matrix protein. So that's very important to realize that KLF15 also plays a role in vascular smooth muscle cells. And one of the models in preclinical that we utilize to do this is known as the femoral wire injury. It's a very tricky surgery to do, but I think that's easily doable. And there's lots of YouTube videos. But when you do that, what you see is that essentially the expression of the KLF15 after injury goes down. So that means that the cells can now, in presence of, when you overexpress KLF15, you upregulate PDGF and you upregulate downregulate proliferation and downregulate migration. But when you knock it out, the opposite happens, right? So it's the same story, that when you knock KLF15 down, it's actually a problem. In the vascular smooth muscle cells also. So that's a different phenotype, but it's the same idea. And this is looking at Briard-Hughes staining of those same intimal lining of the arteries that were injured. Yeah, so essentially the story goes similarly, that in vascular smooth muscle cells, KLF15 negatively regulates a bunch of these players that then turn off the PDGFB. And then that's really important for KLF as a mechanism to control vascular smooth muscle cell proliferation. Obviously, these have huge implications in human disease. And as I already said, there's like KLF15 CAD associated. And so essentially establishing KLF15 as an important determinant of cardiovascular response to stress. This is sort of the overarching picture that I wanted to leave you with, is that the hypertrophic gene up, fibrosis up, hypertrophy down, fibrosis down is KLF15. So you see there are player like KLF15, which turns down these mechanisms. And then there are other players, the yings and the yangs, there are other Kruppel-like members who do the opposite, right? So that's important to note, because KLF15 is not ubiquitous and unselective. It's very selective in its function. And it's most likely because KLF15 interaction partners are very tissue and cell specific. So it's important to know the red is a very selective mechanism, and the green on the right is also a selective mechanism. And each of them, these players, have a very specific, tissue-specific, cell-specific effect. So not every Kruppel is created equal. Turns out that the KLFs are not just important in smooth muscle proliferation, they're also very important in fasting exercise, circadian rhythm. And there's a whole lot of metabolism stories that are coming out of this field that I don't have time to go in. For the fasting, I'll just talk a little bit about it. There are some SNPs identified in certain Kruppels, including KLF15, that are associated with metabolic syndrome. So that's important to note. The KLF15 metabolism initial links show that the KLF15 expression and the tissue types are connected to these kinds of phenotypes. It now so happens that the KLF15 liver is expressed in the liver, in the muscle, and it actually, the pattern is, you notice that there's a diurnal cycle, a circadian rhythm difference in its expression pattern. So it's really up when it's sleeping and it's down when you're awake and feeding, right? So that's an important observation to suggest that perhaps KLF15 also gets upregulated in terms of exercise. Because, in mouse, their sleep life cycle is opposite our, human sleep cycle, obviously, right? So that's an important point to note, that the circadian rhythm of our body is actually directly connected to our, the circadian rhythm is connected to our, the body's, the mechanistic underpinnings. And KLF15 is upregulated in exercise, and KLF15 is upregulated in skeletal muscle in aerobic exercise. That's important. When you create a knockout mouse in adipocyte and KLF15, you see how the mice are very lean. So they are very lean mice. So it means that KLF15 regulates lipogenesis and lipolysis. And these particular factors, give me a minute. What happened here? Apologize. Essentially, the KLF15 regulates lipogenesis and lipolysis. And it's important to note that in the knockout KLF15, basically the mice are leaner and they run faster. The PPR alpha group is another group of transcription factors that KLF15 is a partner with. I don't think I'll have time to talk about KLF2. I may have spoken too much already, but KLF2 exerts also, which is another transcription factor, exerts local control in a broad setting. And has the similar kind of storyline. The interesting thing is that the statins induce KLF2. So that's important to know that, we were thinking about, can we design some super statins that can regulate KLF2, and because KLF2 is a broad setting like thrombosis, obesity, atherosclerosis, and other disease. So superstatins upregulate KLF2, and upregulation of KLF2 downregulates thrombosis, obesity, and other diseases. And can we create a model where KLF2 can act as a biomarker at a very basic level? And so we have some ongoing studies on that. Some of these are published, and you can read that. I'm just going to not talk about all of this because lack of time. But essentially, I'd like to leave you with the idea that a combination of KLF15 and KLF2 and other crupples can give you an understanding of the toolbox that you need to study the preclinical models, and then to kind of translate them, to come up with genomic association studies to understand how these particular pathways are directly involved in the disease pathway. And if they lie in the causal disease pathway, it's an important determinant for the trajectory of the disease. And so if you can regulate each of those proteins, mRNAs, or genes, whatever you focus on, at a very basic level, you probably have affected the outcome of the disease. And the question is, which of these pathways is really important and which of these is not? And a good preclinical model often tells you that very early. Now, just to completely switch slides, one of the things that I'm working on, I just included the slide last minute. This is a study that recently I published in Diabetes. You can look it up. Given the epidemic of diabetes in our country, it's important to establish some, really good therapeutic options that do not have a lot of off-target effects. One of the strongest frontrunners right now is dapagliflozin, a SGLT2 inhibitor. And what we notice is that when we created a rat model of streptozocin-induced diabetes in presence of dapagliflozin, or I call it DAPA, you notice an improvement in all of the diabetic parameters that we look at, and not just in the human population. We had a small human cohort where we did that, and that's all part of that publication. You can look at that. But we also figured out that the mechanism that by which this DAPA, much like the statin story, is working, is that it's working through the ER stress pathway. And the question now is, can this pathway be established as any of the biomarkers, that go up or go down that same theory that I described, like whether a gene goes up or down, sometimes determine the disease trajectory this way or that way. So we're actually exploring this further through the use of dapagliflozin in the context of STZ-induced rats. So I'd like to just sort of sum it all up, saying that the drug discovery paradigm in CVD essentially, has changed and morphed, but the essential conditions remain the same. So you identify a cardiovascular condition, hopefully with a plausible genetic basis, and then you translate it to the human condition. And then you develop robust cell and biochemical assays, and then do more precise drugs and causally connect biomarkers. And important to know that the causally connected biomarkers can be, both target engagement biomarkers, or they can be disease biomarkers, but whether or not they are selective and specific. I'm sorry, there's a word missing there. A selectivity and specificity to a specific disease and a pathway is important to establish early. And the preclinical models are perfect to establish those. Then with that idea that if you have a very precise drug, if you have a precisely connected to your target, to your pathway, then you can probably develop rapid diagnostics, right? And then you can identify risk alleles in the human cohort, human patients, via either sequencing or PCR technologies, and connect the dots, meaning if you have the KLF15 SNP allele that shows to be a risk factor in the humans, of course you can correlate that to your preclinical model and test and validate that theory in at the bench. So it's a back and forth between the human studies of the alleles and the variations via sequencing, and the GWAS, genome-wide association studies, and with the preclinical bench, right? Finally, I really want to put in a plug for the computer models. They've come a long way. The computational modeling is a big part of what I now am involved in and doing, figuring out how to detect the disease risk and the risk association early on would really help you develop better preclinical models. So because the AI really allows us to take the human bias out of the machine learning. I apologize for some of my my misspeaks or early on, or some lack of proper coloring of my slides that often happens in a rush. But I'd like to leave you with the this particular idea that in order to succeed in science, you actually sometimes need 4 Gs, as my boss used to say: patience, ability, luck, and money. And that's actually based on Paul Ehrlich, the Nobel Prize in Physiology and Medicine from 1908. And this is very, very true. All of this research comes at a cost. And in my case, most of my support comes from the Department of Medicine at the Mass General Brigham, the Harvard Medical School. And this is the building that I work in, and this is sort of the general area. These are the pillars where the Harvard Medical School students are trained. And I'd like to thank all of you for being very patient with my slide presentation, and a little bit chaotic at times due to lack of time. I'd like to take any questions anybody has, and I'd hand it over to Shinjini now.
Thank you, ma'am, for this very engaging presentation. And it was not chaotic at all, as you said. So we have a few questions.
Sure.
So the first one is from Shuranjana Haldar. She's asking if there is any RNAi level interference that have been tested in KLF15 low expression.
Yeah, definitely. You can, well, when you create so what happens is we don't need to do RNAi interference in the KLF15 because most of the knockout data is available. But when you have a very low expression of KLF, meaning you're talking about KLF15 endogenous level expression, there are tissues that are expressing low levels of endogenous expression. I'd have to refer to the literature. I don't know what's out there now, but I know that there's so many tissue-specific knockouts available for KLF15, and because it's important, then I'm sure somebody's done it. It's not me. So I can get back to you with a clearer answer. But I would envision that when you have a low expression of KLF15, that's normal physiology, because most of these important markers and genes, they actually don't have to express at very high levels to do their day-to-day operation. They only get turned on, upregulated, or downregulated when there's a problem, right? Because they're sort of the sensor sitting there. The way I like to envision these cripples, they're sensors, and they're sitting there, they're doing their job, and then all of a sudden the problem comes, and they're like, well, we need to act. And that's how you think about these. So a low level of expression of KLF15 could mean nothing, but when you knock it out completely systemically, then you realize you got a whole lot of problems. You got a heart problem, you got a lipid problem, you've got a metabolism problem. So, at the tissue-specific level, you start teasing out those kinds of issues. So I hope that answers your questions, but it is a good point to consider low-level expression of genes. Yeah.
Okay, thank you, ma'am. And another question is from Akashlina Marik is, can blood glucose level affect expression of KLF15?
Yes. This whole, I don't know if you remember the slide I showed. I know I showed very fast, and it'll be available on YouTube for you to look at. And it's also published. The story of KLF15 and GLUT4, the glucose transporter protein 4, is very important. And the fasting and the feeding cycles, the fact that the KLF15 expression is diurnally regulated by the nocturnal cycle, clearly indicates that the blood glucose level itself will affect KLF15, because the fasting and feeding cycles, after fasting, your glucose level goes up. So if you put it on a glucose GTT clamp, which we have a paper out in Cell Metabolism, you can look at that one, and then clearly shows that there is an effect on KLF15 level expression. And so it's important that, yes, glucose itself should affect the levels of KLF15. Thank you for the question.
Thank you, ma'am. So that was it today. So I thank you again for giving this wonderful lecture and agreeing to speak for us. And thank you everyone else. Thank you the audience for joining us. And I'm sorry that ma'am couldn't join today, but I'm sure she'll have interact with you later on.
No, I really appreciate the talk. I realized that I needed to brush up so many of my slides, this is a golden opportunity to do that. But if anyone has any questions, they can reach you, Shinjini, and then you can connect with me anytime. I love to teach. I hope that came across, and I'd love to talk to whoever wants to talk to me about the science.
Yeah, so if anyone has any question, please email us on our seminarsatcu@gmail.com, and we'll forward it to ma'am.
Sure.
So,
all right.
Thank you, ma'am.
Thank you.
This day.
Thank you.
And thank you, everyone, for joining us. Join us again next week on Saturday, the 17th of July, for a lecture by Dr. Shobuj Kumar Chaudhary of University of Calcutta. Please subscribe to our YouTube channel. Our previous talks are all archived there, and you can check them out. You can also visit our website www.zoologyhub.org. Thank you again for joining us. Take care and see you again next week.